Welcome to our podcast on cartilage. We are moving away from our connective tissue proper and moving into our specialized connective tissues. And as we look at the different types of cartilages, you're going to see some similarities, some differences. So this is a nice tissue to compare and contrast. However, you are absolutely going to be recognizing what we see in the way of these nested groups of cells. You might have a lot of them together. You might have fewer of them together. But these larger cells somewhat collectively together in these nested areas, these are typically going to be what we'll see when we're looking at cartilaginous tissues. Now, with cartilage in this unit, we are going to be able to define the molecular components of cartilage. There are going to be some constituents that are going to be specific for cartilage, and we're going to know the composition of cartilages, and we're going to be able to identify the different types of cells present in the different types of cartilages. And when you put those together, the molecular components, the cells and their histology, that's going to lead us to be able to classify the different types of cartilage that we see. Now, we've already mentioned that cartilage is a specialized connective tissue, and this is going to be a structural kind of connective tissue. And cartilage and bone are both related. They're going to be cells that secrete a matrix and surround themselves and eventually get trapped inside this little pocket that we're going to call a lacuna in both of these. We see cartilage here on the left, and we have our bone here on the right. We'll be covering this in a subsequent podcast. So the cells we have in cartilage begin from mesodermal precursor cells, and the cartilage cells, once they have specified themselves for a cartilage fate, they're going to be called chondroblasts. Now, chondroblasts are actively producing a cartilaginous matrix, and they will, in fact, paint themselves into a corner, if you will allow me to use that analogy, and surround themselves by this cartilaginous matrix. The space within which the chondroblast resides is called a lacuna, and once they are completely surrounded, that's going to signal for them to lower their activity, and then they become chondrocytes. So initially chondroblast, fully matured and surrounded, chondrocytes. Now, you might think once you completely surround yourself, how do you have any sort of communication with the external environment, with the vasculature, other cells? Well, cartilage is very hydrated. It is not a solid wall. And so chondrocytes are going to be able to exchange materials by diffusion. Now, with many of our other tissues that we are getting into, and as we build our organs in larger and larger structures, we're going to see the different components that reside within each. And in cartilage, we are going to see that the acellular components are going to be primarily glycosaminoglycans and proteoglycans, which we'll talk about in a bit, collagens, which we've already discussed, but for cartilage, this is primarily going to be type 2 collagen, and then we're going to have quite a high concentration of elastic fibers present in cartilaginous tissue. So in our illustration, we can see a chondrocyte, which means this cell is completely surrounded by cartilaginous matrix. We have our type 2 collagen illustrated here. We can see the large fibers here as well. We have our proteoglycans, glycosaminoglycans, and even this large proteoglycan aggregate that we're looking at right here. So what this is, you have a core of hyaluronan. Another name for hyaluronan is hyaluronic acid. Suspended on this glycosaminoglycan are proteoglycans. Each one of these units, which consist of a protein core on which are covalently attached glycosaminoglycan chains. This is a proteoglycan. In this case, these chains are primarily chondroitin sulfate, and the composition of the protein, its primary sequence in the chondroitin sulfate, these are aggrecan chondroitin sulfates that aggregate together in these large complexes. Now, they're highly sulfated, so that negative charge on those sulfate groups is going to help sequester water within this tissue. Now, with all of the water sequestered in this area with the elastic fibers, cartilage is going to be a great shock absorber. We're going to find this on places where we have bone-on-bone contact. This is also going to enable us to have a friction-free surface rather than have bone-on-bone rubbing together. And you may see patients down the road in your clinics that have arthritis, rheumatoid arthritis, where the cartilage has been lost from some degradative disease, and you get bone-on-bone contact, and it's very painful. But this low friction surface, and cartilage is not going to have a lot of blood vessels and nerves in it to begin with. This is going to enable those joints to move very, very freely and be pain-free as this moves. Now, cartilage is going to be tied in with our supportive interstitial connective tissue because we somewhat have a transition zone between our cartilage and our external interstitial connective tissue. The perichondrium, this is the area that surrounds and forms the outside of our cartilaginous tissue, is comprised of two layers. The outer layer of the perichondrium is basically fibrous connective tissue, and you have blood vessels present in this as well. But the inner layer of the perichondrium, which is now going to be a cellular layer, we also call this a chondrogenic layer because we're going to have chondroblasts undergoing mitosis, and so we're going to have growth from the perichondrium out. This is going to be our inner cellular layer of our perichondrium. So as we look at this light level microscope, you can see this is somewhat of the boundary zone. We're looking at a very close-up view of the perichondrium. Here you can see our collagenous fibers. This is going to be our outer fibrous perichondrium. And as we move deeper into the cartilage layer, this is our inner cellular perichondrium before we get into the cartilage tissue proper. Here we can see the lacuna and the chondrocytes that are embedded within our cartilage cells. So we have our molecular components. We have our cellular components. We're being able to recognize them. Now we're going to classify these tissues based upon those constituents, the acellular constituents, and the histology and the cytology, the looks of the cells within each. So our three types of cartilage. The most extensively that we find in the body is hyaline cartilage, and that's here on the left. We also have elastic cartilage, and many times when you see elastic cartilage stain, it's going to be this darker black color, and you're going to see all of these fibers. These are the elastic fibers stained with this very specific stain. And the third type of cartilage is going to be fibrocartilage. Now, when you first see fibrocartilage, you are going to be hard-pressed to distinguish, is this dense, regular connective tissue like a ligament or a tendon, or is it fibrocartilage? Because they're really going to kind of occur in the same place. But what we're going to be looking for that's characteristic of our cartilage are these lacuna, these spaces within which these cells reside. And in many instances, we are going to have cell division after the lacuna form. And so several cells are going to be present within one lacuna in these isogenous groups or cell nests. We can see that we've got two chondrocytes in that lacuna, two there, two there. So we have these nested structures, and that's going to be very characteristic of all of our types of cartilage. And we'll be looking at those as we move through these types. So first, hyaline cartilage, the most abundant cartilage we have in the body. It's all over. Our nose and larynx, ventral ends of our ribs, the tracheal rings in our airway, and even our bronchial rings are supported by hyaline cartilage. Many of our movable joints, like our knees especially, but also our elbows, are going to have these articular surfaces. These are present in synovial joints. And then we have these locations within our long bones that are called the epiphyseal plates or growth plates. And it's the cartilage that enables these bones to lengthen. And we'll be spending a lot of time talking about these growth plates as we get into the lengthening and growth of bone. But that's going to be in the next podcast. Now, when we look at hyaline cartilage, and again, we'll do this in our laboratory podcast as well, you're going to see this light staining in most of the areas, but then you're going to see these really dark patches, really basophilic staining to the matrix around many of these cells. You're going to see the cells, they're not packed so tightly together, and in many instances, you're going to see these cells appearing in groups of 2 or more cells together. These are isogenous groups. Here's some cells together. And this highly basophilic area surrounding the cells, this is going to be part of their territorial matrix and is very characteristic of hyaline cartilage. Here we can see a perichondrium. We've got our fibrous perichondrium. We've got our intercellular perichondrium. And then we get into our hyaline cartilage proper. So hyaline cartilage, like all of our other cartilages, start as chondroblast, laying down their cartilaginous matrix. They start as mesenchymal cells, which are derivatives of our embryonic mesoderm. And they're going to form these groups. Now, these groups form much like what we're seeing here. You see these two cells within this one lacuna. Here we see another. These are cell nests or isogenous groups. Once a chondrocyte, chondroblast, forms its lacuna, if the cell undergoes division, there's no place else for that daughter cell to go. So they stay within the same lacuna. That is characteristic of our hyaline cartilage, but also characteristic of our other types of cartilage as well. Now, in this image, you can also see this dark area around our lacuna, much darker than the areas further away. This is our cartilaginous matrix, and the matrix closest to these lacuna, closest to these isogenous groups, that's going to be our territorial matrix that we'll see. It was the dark purple that we saw in that previous light microscopic slide. Now, we mentioned the proteoglycans, but collagens are also extremely abundant in cartilaginous matrix. In fact, type 2 is the most abundant, and for decades it was thought type 2 collagen was only found in cartilage. We know we find it in other areas, but I still think most textbooks will say that type 2 collagen is the cartilaginous collagen type. And if you look at the dry weight of cartilage, it means you take all the water out, then 40% of the dry weight of hyaline cartilage is going to be type 2 collagen. So that really speaks to the quantity, the vast quantities of type 2 collagens that's present in the cartilaginous matrix. Here we see this illustration. We've spoken about these before, but our proteoglycans, and the most abundant is going to be aggrecan, a chondroitin sulfate, which is the glycosaminoglycan chains that you see here in red. These are our chondroitin sulfate chains attached to our aggrecan core protein, which then can organize itself into large aggregates along with the blue line that we see here, which is hyaluronan, also known as hyaluronic acid. It's extremely long, high molecular weight glycosaminoglycan itself. It doesn't have any protein that is covalently attached to it. It just organizes the proteoglycans around it. Those sulfates, again, are so important for the attraction of water into this tissue to keep it very hydrated, which creates that hydrostatic pressure to resist those compressive forces. And like collagen, type 2 collagen accounted for 40% of the dry weight of cartilage. This sulfation and the aggrecan that's present in cartilage accounts for 80% of the wet weight. So those are the two principal components of cartilage, the type 2 collagen and our chondroitin sulfate proteoglycan aggrecan. Now, within this matrix, we also have to have a way for the cells to adhere to the underlying connective tissue and the extracellular matrix. And much like we saw laminin for epithelial cells and fibronectin for fibroblasts, chondronectin is an adhesive glycoprotein for cartilage, and that's going to enable it to anchor and bind to type 2 collagens, chondroitin sulfate, hyaluronic acid via some of the integrins that are on the surface of our chondrocytes and chondroblasts. Now, in this illustration, it's a little bit too high magnification, and as you pull away, you can see the territorial matrix a little bit better. This arrow isn't exactly pointing to the territorial matrix. It's pointing to the lacuna around this isogenous group. But in some areas, you see a light stain, and as you get closer to the cells, do you see how this gets darker? This is going to be our territorial matrix, and on average, it's about 50 microns away from the edge of these isogenous groups and away from the edge of our lacuna. In this illustration, again, we can see this purple shadow that's in the near vicinity of our lacuna. and this is going to be our territorial matrix. And as we move further away, you can see this lighter matrix in the cartilage. This is going to be between our territorial matrices, and so it's the interterritorial matrix. More collagen, fewer proteoglycans, and those negative proteoglycans are really hanging on to this stain. That makes it nice and basophilic. Now, again, looking at the same image, we have our lacuna within which the cell resides, but then we are also going to find a pericellular capsule. And what's going to happen is we have a layer that is in between the edge of our lacuna, which is going to be our cartilaginous matrix, and the plasma membrane of the cell. And within this, it's going to basically be similar to what we see with the components of the basal lamina and basement membrane, on which the chondrocyte is going to be able to attach. But this is also going to help protect that cell against the mechanical forces that are happening in cartilage, because imagine the compressive forces from your body weight pushing down on your articular joint of your knee, or the stresses of your elbow, your shoulder, your hip, in these places. So we have to have protection for these cells against these forces, and that's the pericellular capsule that's able to do that. Here in this illustration, C is trying to point out the pericellular capsule, but you may also think that this is pointing out the edge of the lacuna. So in this case, you would need to be aware of the question that you are being asked to be able to appropriately identify this area. If you were asked, what is this space within which the cell resides? It's the lacuna. But if you were asked the components of, let's say, the extracellular matrix that reside along this edge, what would that be? And that's going to be the pericellular capsule. Now, cartilage is going to respond to a lot of the same hormones and factors that stimulates growth. And so in this case, thyroxine, testosterone, somatotropin, those stimulate growth and certainly are going to stimulate the growth of cartilage, especially as we look at developing bone. Because in many instances, such as our long bones, we first lay down a cartilaginous template, and that is later converted into bone. Also, cortisone, estradiol, these are going to actually slow down and inhibit cartilaginous growth. Now, in many instances, when we do not have sufficient materials, either through our diet, as we see here, vitamin D is extremely important, because if we don't have enough vitamin D, we are going to insufficiently calcify our hyaline cartilage. That's the step in the formation of bone. And if we do not sufficiently calcify, then we are going to have weakened bones. They're going to bow out, and that is a typical textbook symptom of the disease known as rickets. Okay, moving to our second type of cartilage. This is going to have some of the same characteristics that we see with hyaline cartilage, and yet differences. And certainly in location, these are going to be places where we don't necessarily resist compressive forces, but we need elasticity and a rebounding. So the pinna of the ear, external and internal auditory tubes, the epiglottis, the larynx, these are typical locations where we will find the second type of cartilage, elastic cartilage. Now, we call it elastic cartilage because it is much more rich in elastic fibers than what we saw with hyaline cartilage. We're still going to have type 2 collagen. We're still going to have our isogenous groups. But for me, when I look at elastic cartilage, especially if it is stained for those elastic fibers, I see many more cells in elastic cartilage than I did in hyaline cartilage. There's just an abundance. And these are much more compacted together, much more randomly distributed. And again, with stains for elastin, you can very easily see these fibrous components running through the matrix of elastic cartilage. You don't see these fibrous components typically when you're looking at hyaline cartilage. Our third type of cartilage, this is fibrocartilage. And with fibrocartilage, we will have some type 2 collagen, but now we're going to begin to see a lot of type 1 collagen. And this is what really gets this tissue histologically confused with looking at dense regular connective tissue, because you're used to looking at this nice reddish look to our collagen staining. However, as we look at the cells that are present here, you're seeing lacuna present in these. You're not going to see fibroblasts looking like this. Fibroblasts don't typically look this rounded either, and as you look, we may have an isogenous group. May have some isogenous groups in here. So you have to look very carefully as you look at these different tissue types, because fibrocartilage and dense regular connective tissue are lookalikes when it comes to histological preps. So fibrocartilage, where do we find this? Our intervertebral discs, our pubic symphysis, attached to bones, and this is going to be sort of a transition point where we're attaching our bones to hyaline cartilage and attaching hyaline cartilage to our ligaments. So you're going to see these in all the same places. And when you see hyaline cartilage, almost be expecting to see these fibrocartilages linking into our ligaments or linking into tendons even. So we have chondrocytes, but in this case, the chondrocytes are derived from fibroblasts. Now, fibroblasts are the cells that lay down type 1 collagen, and we already said there's an abundance of type 1 collagen present in this tissue, so it's not surprising that these are present from fibroblasts. However, these chondrocytes do not look like fibroblasts. They're not as elongated. And in many instances, we will see some of these isogenous groups. We'll see these lacuna, but these are also going to be aligned in parallel rows, as opposed to seeing the fibroblasts scattered throughout our dense regular connective tissue. So really, your objective here is not to distinguish this from the other types of cartilage. That's easy. Here, we're trying to distinguish this from that dense regular connective tissue that I keep harping on. Here, as we look at a sample of cartilage, even at this magnification, I think you can begin to get an appreciation that this is hyaline cartilage. We can see our isogenous groups. They're not as packed tightly together as we would see with our fibrocartilage or elastic cartilage. Here we can see our perichondrium. The fibrous component is actually detaching, and we can see fibers tying in our underlying tissue with our perichondrium, that outer fibrous perichondrium. And our cartilage is going to grow in one of two ways. We can either have interstitial growth, which is the growth from within. That's what interstitial means. And this is the formation of those isogenous groups, the cell division occurring within each of those lacuna, versus our appositional growth, which is the growth from the outside. So we're adding new cartilaginous material. We're adding new chondroblasts from the cells of the inner cellular layer of the perichondrium. So two ways that cartilage can expand: from within or being added to from the outside in the perichondrium.